mutation rates vary across many different types of organisms, and generally speaking, mutation rates include, as we've seen, both the beneficial and neutral and deleterious mutations. Generally speaking, a large portion of mutations are neutral or deleterious. This is showing results from a couple of different types of organisms, a virus and a yeast. What they are looking at here is the fitness of the relative compared to the ancestor. So if you compare how fit the ancestor is to, say, the descendant, if the value is 1.0, if that's the ratio, it means there's no difference. The descendant has the same fitness as the ancestor. Anything less than 1.0 means that the descendant, the mutated individual, has less fitness. So if you look at these bacteriophages here, you have a certain percentage, about 20% of the mutations were lethal. Some of them were extremely deleterious. A fair number were slightly deleterious, and only a small percentage were beneficial. And something similar is seen in the example of the breeder's yeast there. Another thing that has been learned since we have access today, of course, to looking at sequences of DNA, which wasn't available decades ago, is it turns out that genetic variability is often a lot higher than was once thought, because there were many things that weren't expressed. And if all you could see was what was expressed in a fruit fly, you would assume that genetic variability was a lot less than it actually is. And in fact, genetic variability, diversity, can often be fairly high, particularly in large populations. That was essentially all that I needed to say there. There are a few other things we might have covered if we hadn't lost half a week, but that summarizes a little bit about mutations. So this fourth topic relates, in fact, to chapter, I think it is chapter 4 of your text, but I'm not positive about that, about phylogeny. Phylogeny and phylogenetic reconstruction. This is not only relevant to your first test, but this gives some of the background information that's relevant to the first phylogenetic exercise. Somebody pointed out the other day that first time I put that on Moodle, I had neglected somehow to get the last 2 pages on there, but I fixed that. It should be complete now. There were some organisms that were not on there. So the first phylogenetic exercise is available. That can be done individually or in teams. And there was a due date on it. Don't remember off the top of my head, but it's listed. All right, so this is on phylogenetic reconstruction and what phylogenies are. The goals of this type of phylogeny is to basically construct the evolutionary history of a group. So when people say, as we said earlier in another lecture, that fungi are more closely related to animals than they are to plants, that's basically a phylogeny, a phylogenetic relationship. Evolutionary trees are phylogenies. So we try to construct these using our data. All phylogenies are hypotheses, because just like any other hypothesis, they can be tested, and new data can sometimes change our thought on relationships. Now, it may be an hypothesis that raccoons are more closely related to dogs than they are to trees, but that's an hypothesis I don't think we need to worry about is going to be overturned. Other hypotheses, of course, which are more tenuous, may be altered. A lot of classifications that I learned at one time many years ago have changed as we realize with new types of data, new techniques, that relationships may be different than we thought. So the goal is to construct an evolutionary history using the best data available, and it is essentially an hypothesis of how things are related and what their history is. various methods have been used, but the dominant method today is essentially one called cladistics. You will see the term clade in several contexts. Clado, which might be clade or clado, various terms, comes actually from a classical Greek word referring to the branch of a tree. The idea of an evolutionary tree as a way of visualizing relationships goes all the way back at least to Darwin. He used the tree idea. And so cladistics is the science of forming these branches and putting together in a tree according to a particular methodology. I'm not, of course, a practicing cladistics person, but I do see the results a lot, and it is a fairly common technique. The previous person who taught this course, Professor Nealon, was actually a cladistics individual. He did this sort of thing for a living with plant groups, and not too surprisingly, his cladistics coverage took several weeks of the semester and an awful lot of exercises. We cover it here, but maybe not quite as much. We all have our different approaches. So we'll take a look at what the methods are, but there's a first things we have to do is look at what types of characters are used. What data do we have to use to construct phylogenetic trees or evolutionary trees? Well, we have to base it on traits or characters. They're often called characters in this type of work. Characters do have to be heritable. If the character isn't heritable, then of course it's not going to tell you anything useful about evolutionary history. Now, a character could be a body part, some kind of anatomical characteristic, or a cell structure. For an animal, it even could be a behavior, as long as the behavior's heritable, which often is. It could be something in the physiology, a type of physiological pathway, a biochemical feature. All of those could be characters. And of course, since the 1970s, and today extremely heavily used, of course, are molecular sequences. Originally it was amino acid sequences because they learned how to do that first. But of course today DNA sequences are used extremely heavily in cladistics. So those are characters as well. In addition to being a heritable trait, the character to be useful has to indicate relationships. What types of similarities among organisms occur because of common ancestry? Is it the homology or the homoplasy? The homology or homology is, remember, those are traits like the forelimbs that we talked about that are similar because of common ancestry. And common ancestry shows relationships. So the types of traits to be used ought to be ultimately relate to homology. Now, there is a bit more to that than simply homology, but at least as a starting point, you need homologies. The homologies could be, as you see, it could be anatomical, or it could be homologies in gene sequences, but it has to be an homology to be useful. Homoplasy is something we like to minimize. Homoplasy, let's see, I don't have a picture of homoplasy right there. Homoplasy includes convergent evolution, what we sometimes call the analogy. Remember, these are the traits whose similarity is not because of relationship, but because of similar function or adaptation. Those are fine for studying some ways in which evolution works, but they don't show you that things are related to each other. As the wings of a bird and the wings of a bat do not prove that birds are more closely related to bats than, say, bats are to rabbits, because those are simply convergences. Another type of homoplasy is a reversal. This is a character that changes and then returns to its original state. True reversals are certainly seen in DNA mutations. It's perfectly possible for a C in a particular position in the DNA to undergo a point substitution, become a T, and then later on, become a C again. That's a reversal. That's also considered a homoplasy. And traits that undergoing that are also ones that one would prefer not to use, or in those cases might not even be recognized sometimes. So these are the sorts of things we would prefer to avoid. Now, of course, recognizing whether something is a homology or homoplasy is a whole different story, but at least in principle, we want to use homologies. We also want our characters to be independent of each other. It's usually we prefer to use more than one character because the more characters you use, the more confident you are going to be in your results, just like any kind of statistical test. When I say that they're independent, you can't have a situation where if one changes, the other changes, because then you're really not looking at two different things. For a crude example, let's say one of your characters was the volume of a bird's beak. Would another character that would be legitimate to say your second character is how deep the beak was? Or does the volume of the beak depend, among other things, to how deep the beak is? Yeah, those aren't independent of each other. Or you wouldn't use the volume of the beak and the length of the beak, because those are related characters. Characters have to be independent. You could use the volume of the beak and the length of the toe claw if you wanted to, if that were relevant, because those are related to each other. So you need independent characters. Sometimes that's not always easy to recognize. That was a crude example I gave you, in which case one might be misled in terms of your confidence in a result. The science of cladistics was essentially founded in its modern form by Emil Hans Willi Hennig, and you can blame him for all of the types of terminology we use. He is considered the founder of cladistics or phylogenetic systematics, which is the dominant method used. And as part of that, he created the terminology used, which can be confusing, simply because there are a lot of rather unfamiliar words, as we will see. We have plesiomorphies, apomorphies, synapomorphies, paraphyles, polyphyles, and monophyles. This is simply the nature of the beast, and it does need to be the sort of thing you're going to have to work through. I will say your textbook actually has a very good descriptions of these, and it's well worth reading through those as well as looking at the lecture. So we'll start out with looking at the structure of a tree. This is a phylogenetic tree. Another jargon term for something like this, if it's formed using cladistics, it's called a cladogram, which means an evolutionary tree, a phylogenetic tree. They're often called cladograms. All right, so this happens to be of a number of cat species. You can see we have Canada lynxes, bobcats, tigers, jaguars, jaguarundis, if you know what that is. A number of different species of cat. Now, the particular set of characters they're using are the coat. In particular, the type of spots, stripes, or other features of the coat. Now, is this the only feature that has ever been used to classify cats? No. So we're going to simply look at this as if this is the only evidence. And what has been done using this is they did a phylogenetic cladistic analysis to come up with some relationships based on that. But let's look first at the structure of the tree. How a tree is shown varies a little bit from one source to another. I kind of like this approach, where each branch point is really clearly, easily distinguished. However, there are actually a lot of different ways you see these. These all show exactly the same branching pattern. I don't see this type too often. I don't even know why those are there. I hate these, but these are actually quite common, particularly when you look at things like all species put together. It shows the same branching pattern. It's just, for me, it's never as obvious to work through it. But you do see those a lot, like when they show you all of the things going back to the Archaeans, all the relationships, they'll do it that way. This is also not one of my favorites, but it's very common, some variation of this, because apparently this was easy to generate in printing computers and it uses less space, so you do see these a lot. All of these have exactly the same information, they're just presented in different ways. But I like this one. Besides, this is the one your textbook has all the pictures in. All right, so if we take a look at this, let's look at some of the terms first. You notice we have all of these little filled in dots with numbers. Each of those is a point where you see a branch. One branch forming into two branches, in most cases, a branching, a split. Those are called nodes, as you can see here on the left. Nodes are the points where you have a divergence. So every time you see a split in a clade, it's a node. All right, so another term that's useful here is a clade. Well, clade means branch. Basically, after each node, there's a clade. All of the branches that come after a node are the clade. It might be a single line. This is a clade that leads to the Canada Lynx. But all of this is a clade coming after this point. Because if you think about the branches of a tree, you might have a branch of a tree that is truly simply one branch, but if a whole massive limb fell off, it would have lots of branches coming off it. They're all clades. You can have a clade within a clade. So the clade is whatever comes after the node. So lots of different clades in here depending on where you start. So nodes and clades are part of our basic architecture, the way in which this shows relationships. The confusion, not so much confusion, but some of the difficulty of the terminology goes back to how we represent certain types of evolutionary relationships. Transitions are new evolutionary features. Sometimes they have been called evolutionary novelties. A new character. These are called transitions. This is a different use of the term than transitions in mutations, although they're not totally dissimilar, but this is anything new. A bobtail, for example, is a transition. Transitions are indicated by these little slash marks. What this says is that here we have node 1, and it's split this ancestor, hypothetical ancestor with a flecked coat, because we don't actually have that ancestor. We have first two different lineages appearing. In one lineage, they remained similar to the adult with respect to the ancestor, I should say, with respect to the tail, which is a long tail. But in this other lineage, in this reconstruction, at some point, you have the evolution of a shorter tail, a bobtail. That's why bobcats are called bobcats, because they have short tails. Lynxes, there's actually another couple other species of these, have short tails, which is different than other cats. So at some point, after this divergence, in this clade, they became bobtailed. So both of these still have bobbed tails. That's a transition, an evolutionary novelty. Now, there could, of course, be more than one evolutionary novelty along one of these branches, but there's going to be at least one. So here we have two existing species in North America. Their most recent common ancestor will, of course, had a bobtail, so they have bobtails. Notice they use the word sister taxa. Those are also called sibling, depends on which word you want to use. These are two groups, could be species, order, whatever they are, that are the most closely related to each other. There are no brothers. They're always sisters or siblings. I have no idea why. That's why they're only daughter cells. You ever have sun cells? It's probably all Victorian in some way. All right, so here we have the bobtail. That's a transition. That transition, of course, did not occur here. That's an evolutionary transition, a new evolutionary character. All right, so those are some of our concepts. Now we have to get into Willy Hennig's various terms, apomorphy, plesiomorphy, and synapomorphy. I won't even use symplesiomorphy, that's too weird. All right, so what is an apomorphy? It might not help much, but I'll tell you a little bit about where the word comes from. Morphe is just referring to a character. Apo, in this Greek form, is more or less applying from, away from. An apomorphy is any transition that occurs after a particular node. It always has to be in reference to a particular node. So anything that comes after a node is an apomorphy from that point on. So if this is node 1, all of the transitions here are apomorphies with respect to node 1. However, if you go to node 2, then the bobtail is not an apomorphy of node 2. But rosettes, a different form of the spot, so to speak, are. So are stripes, because stripes are coming after node 2 within this clade. So basically, an apomorphy is any transition, any evolutionary novelty that's coming after a particular node, after a particular branch point. So within the clade, it's an apomorphy is after the branch point that formed the clade. That's known as an apomorphy. Now we also have, however, homologies, which occurred before a branch point. So if we look here at rosettes, or actually even better than rosettes, let's take a look here at stripes. All right, if we look at stripes, stripes are an apomorphy of node 4 because the ancestral rosette, according to this reconstruction, within the lineage that led to tigers, changed into stripes. All right, so they're an apomorphy of the tiger's clade. Rosettes are not an apomorphy of this clade, but they're still homologous. The more ancestral transitions that are before in ancestral history, before the node, are called plesiomorphies. They are more basal. Sometimes they're also called primitive, although that's not a particularly good term. If you were in 102, we might have called those primitive shared characters. Apomorphies are also called derived, and that relates to the meaning of the word apo. They are derived in the sense that they derive after the node. So plesiomorphies are homologies that are before the node, and apomorphies are the ones that come after the node in a particular branch or set of branches. Earlier in the course, I just threw this in because we had covered it early, I did actually put a few of these things on here. Remember this one about looking at the middle ears? These slash marks are actually transitions. So one might say that some bone that we could change, whatever names they are, the basic bone here is occurring here as a transition. The stapes is a form of it, is a transition that applies to this node, so it's an apomorphy of this node. But if you look at all of these, its hyomandibuliform is a plesiomorphy of that. All right, so far we see then that there are apomorphies and there are plesiomorphies, or often called basal or derived traits. Derived traits being apomorphies and basal traits being plesiomorphies. All right, so to make things more clear, we also use a term called synapomorphy. Actually, this probably doesn't make it more clear, but that's the way it is. Synapomorphy. A synapomorphy is an apomorphy, but a synapomorphy is unique to a particular clade and separates it, distinguishes it from its sister group. We can show that in an illustration over here. Stripes are an apomorphy from 4, and stripes distinguish them from the most closely related cats. So it's a synapomorphy of this group. So a synapomorphy has to be unique to the particular taxon or clade we're looking at and has to distinguish it from the group to which it is compared. The distinction between synapomorphy and apomorphy isn't all of that sharp. More often than not, you see the word synapomorphy than apomorphy. So the synapomorphy is specifically saying it's the characteristic that is unique to the group that separates it from, say, some other group that distinguishes it. We don't have that one anymore. All right. So just to summarize, looking at some of this, I realize there's a lot of unusual terminology here. We have a phylogenetic tree called a cladogram. Each branching point is called a node. Everything that comes after the node is a clade. Clades can be of different sizes. A clade could be a single twig, or it could be a whole massive branch with many branches. After all, mammals is a clade. So is bobcat. Obviously, bobcats are a clade within mammals. We have transitions within these branches that represent new evolutionary characters or new forms of characters. If they're the ones that are coming after the node, they are known as apomorphies. If they are before the node in question, those are plesiomorphies. And the synapomorphy is a character, a particular apomorphy, which can be used to distinguish a taxon, a clade, from its sister group. Sisters are taxa are the most closely related to each other. And we'll take a couple minutes break there to let you digest all of that fine. Cladistics. I think probably a lot of the trouble is just the terminology they came up with. We have in-groups and out-groups. Sounds like some sort of social thing.